So we all start life as a single cell, as a fertilized egg that goes on to divide and divide and divide and divide until we reach adulthood, hopefully, whereupon an adult human has something like 37.2 trillion cells of our own, not leaving aside microbes and so on. And potentially any of those random selections of adult humans have around over 200 cell types in the body, and so they have very different functions, very different morphologies, but they all have the same DNA fundamentally and the same genes. The difference is how those genes are expressed in all those different cells. And so to understand health and disease, we'd like to understand the molecular behaviours of these cells in great detail. And we can do this now from the last couple of years by combining really high throughput DNA sequencing technologies with microfluidics and really smart chemistry, so we can, instead of requiring tens of thousands of cells to sequence a person's DNA, we can do DNA sequencing on individual cells. And so it's worth reflecting on where this came from. And so in June 2000, Bill Clinton, in the last few months of his presidency, hosted this announcement at the White House, announcing the first complete draft of the human genome. So this had taken about 10 years to that point, and when it was finally completed, the first human reference sequence mapped out the letters, the 3 billion letters that make up our genome. It took about 10 years and it cost about $2.7 billion US, so almost $1 per letter. Since then, the decrease in the cost and the speed and the increase in speed for DNA sequencing is just staggering. So if you were to have started the same sort of project in 2001, to sequence a human genome would have cost about $100 million. In 2008, it would have cost about $1 million. Today, we could sequence the individual genomes of everyone in this room in less than a week for less than $1,000 each. So this is just incredible pace of technology. And so not only DNA sequencing, nowadays we can also measure all these other what we call sort of genomic layers of information. So we can measure epigenetic marks that go on top of the DNA. We can look at gene expression across all the genes in the genome. We can look at protein expression and all sorts of other phenotypic traits associated with them. So we're getting really big data sets, and this allows statisticians like me to pretend we're biologists and come and play with the data. Because when you have lots of data, there's a really good opportunity to make terrible mistakes. So you need careful statistical thinking to try and get these analyses right. And this is my favourite stats joke as far as that goes. So why would you want to do this at the single cell level? Well, if you consider cancer, you might start off with a single mutated cell with a change in DNA that then grows and grows and grows, expands. As it grows and expands, it picks up more mutations, more changes in the DNA and its gene expression. So in a single tumour, you have a huge diversity of cell types. And that's what we want to unpick in lots of different ways. And so technologies like this microfluidic chip, which is about half the size of a credit card, hides these tiny little wells on it, each of which can contain a cell, and those little wells containing the cells allow us to do the chemistry on a really small scale to pull out the DNA or pull out the RNA so we can study gene expression. And we can do that on hundreds of cells at once. The next step in these technologies is using things like these drop-seq technologies, so using droplets of oil in water that each contain a cell with all the necessary reagents to do the DNA sequencing again. And so this is already scaling us up from hundreds of cells in an experiment to thousands or even tens of thousands. And so what we want to do is to use this to get underneath the sort of level of tissues and to look at the individual cells, what's going on in these tissues, and looking at the cell types. So it's like, instead of just getting, say, polling data on a statewide level saying we know the average voter, or 50% of the support was for Hillary or whoever, it's like being able to sit down with 1,000 of these voters for an hour and really interrogating them, really asking them what's going on, what's in their minds. That's what we want to get at with this sort of level of resolution. So I'm going to give you one example of a paper published last year looking at all the different cell types of the retina, which is really complicated, going from the cells that detect the light coming into the eye to those that transmit the signal to the brain. And so what these researchers were able to do was to take 45,000 individual cells and, by just looking at gene expression, produce a map like this of all the different cell types. And what they were able to do was replicate all of the known cell types you can look at under a microscope, but also start to uncover new types of cells that you can't tell the differences between just based on what they look like. And so this sort of technology is what we're trying to apply in this HIPSCI project, which is the induced pluripotent stem cells. And so this is where you can take a skin biopsy, so a skin punch from an adult, and reprogram those cells to revert them back into a stem cell-like state from which they can go on to produce any type of cell that you'd like in the lab. And so the thing that we're interested in with our biological collaborators is really early development. So this process called gastrulation, which produces 3 distinct parts of the embryo, the mesoderm, endoderm, and ectoderm. And so an experiment setup we're doing is taking these iPSCs and developing them towards endoderm, which is this layer that goes on to produce the gastrointestinal tract and other important parts of the body. And so to give you an idea of what we actually do in real life, is we have a sort of 3-day process of producing endoderm from iPSCs, and each day we're going to sample hundreds of single cells from dozens of individual donors with individual genetics, individual DNA, sequence the genes, and hopefully understand what's going on. So to give you one idea of what I do on a daily basis, this is what I stare at. This is the outcome of 100 billion statistical tests, and each of these dots on this plot represents an association between a change in DNA and a change in gene expression. And it's these sorts of things that we use to try and understand how genetics affects the behaviour of cells and so on. The end game for iPSCs, where we want to go, is to take those cells that can produce just about anything and differentiate them into all the different cell types that they can produce so that we can study them. And so we want to do that to look at models for drug discovery, how we might be able to correct defective genes, and finally how we could use those stem cells developed from an individual to change the treatment for that individual themselves. The big challenge, of course, is integrating all of this into healthcare. I'm pleased to take one example from my hometown, how Melbourne is starting to think about how we can use genomic technology to actually improve outcomes in the clinic. So we're only started this big project, but this is where it's going. So for me, obviously I'm sold on this. I spend all my time working on it. I think certainly as a research tool, we're already here. This technology is already benefiting Australia. There are researchers in Australia already studying using single-cell technologies and genomics approaches to look at biology. And as I mentioned on that last slide, I think the big challenge for us to consider, which involves the scientists as well as the doctors, economists, policymakers, is how we set up healthcare systems to take advantage of these scientific discoveries and the things we can do from a technological perspective, how we apply them to make changes that benefit people. Thank you.